Hi, everyone. Thank you for checking out my video today. I'm Julia Owen Ramos, and I'm a PhD candidate at the University of California, Davis in the graduate group in ecology. My research primarily focuses on using DNA to study wildlife for conservation and management. And today I'll tell you a bit about my research titled Use of Whole Genome Sequencing to Investigate the Evolutionary History of Island Spotted Skunks. The island spotted skunk is a small carnivore that lives on the Northern California Channel Islands. It currently inhabits Santa Rosa Island and Santa Cruz Island. Island spotted skunks are a subspecies of the mainland western spotted skunk. And in terms of morphology, island spotted skunks and western spotted skunks are so similar that mammalogists once considered the island spotted skunk to be a weak subspecies. However, genetic studies have begun to build a case that the island spotted skunks are more diverged from one another and from the mainland than traditionally thought, and we wanted to further investigate. So we wanted to know, how differentiated is each population? Do island populations show signs of inbreeding? And when did divergence begin to occur? To answer these questions, we performed whole genome sequencing on 2 samples from each island and 2 samples from the mainland. And I'll jump right into our results. First, we used genome-wide FST, which is a measure of differentiation due to genetic structure. And we determined that all populations are highly differentiated from one another. Next, to measure inbreeding, we looked at runs of homozygosity. Runs of homozygosity are long segments in the genome where the pair of chromosomes are identical by descent. When we added up all the runs of homozygosity in each genome, we found that the island individuals had cumulatively more runs of homozygosity than mainland individuals. This means that island individuals accumulated these runs of homozygosity over some unknown period of time while they were inbreeding. And to begin answering the question of when divergence began, we looked at historical demography. We used pairwise sequentially Markovian coalescent, or PSMC, to measure the changes in effective population size over thousands of generations. We saw a demographic split between the 2 island populations and the mainland beginning around 20,000 years ago. However, because each island population may have experienced parallel demographic histories after the split from the mainland, we really lack the resolution to understand when each island began to split from one another. So our next steps, among many, will be to continue estimating divergence times and eventually place the island spotted skunk on a time-calibrated phylogenetic tree. I'd really like to thank my collaborators, and a special thank you to the ARCS Foundation for helping to fund the sequencing costs for this project and for the projects that stem from this research. Please don't hesitate to reach out to me with any questions, and thank you so much for your time.